say that it is wonderful to have this meeting. Kudos to the organizers, Thomas and Mathis. I think you put on a fantastic symposium. I so wish we had been together, together in person, and hopefully we'll be able to do that again very soon. My topic is a very small topic. It's mechanisms of SCAs, models, and compound screens, and I have about 6 hours to discuss it, so bear with me. These are my disclosures. Because it's such a short talk, I think we need to really cut to the chase and say that mechanism studies over the years have led to some overlapping and important general themes of what's occurring in these diseases. And here are 6 major categories of shared mechanisms, or at least overlapping mechanisms we talk about. And of course, repeat expansions are central to so much of the SCAs, and that's a good and a bad thing. I mean, individuals who have a specific repeat expansion, everyone has that particular expansion. But the reality, and I think it's clear, is that this leads to fairly complex mechanisms, and the principle of parsimony may not apply much anymore. This is, of course, a schematic of a gene showing just some of the various neurological diseases that are caused by repeat expansions. And what you can see highlighted in the pale yellow are the SCAs. This is not a complete list. You don't even see, in terms of the recessive ataxias, canvas is not on here, but you can see a number of the ataxias that are. And of course, the CAG repeat expansions, which encode glutamine, the proteins, and hence called polyglutamine disorders by many, are one of the major targets of this SCA part of the global SCA meeting. And I would say that in general, the polyglutamine diseases bring together 3 of these themes quite importantly. One, of course, repeat expansions. And I think the idea that there's impairments in protein homeostasis and RNA homeostasis, and you cannot speak about those as separate things anymore. RNA and protein homeostasis overlap in a very important way. Transcripts are translated into protein, and we're seeing lots of ways in which there's overlap. I don't know if my slides are automatically going as they are, but I'll have to go with them. One thing we know that occurs with the polyglutamine disorders is the fact that the mutant proteins with the expansion tend to aggregate. We've known this for decades, and this is simply from SCA3. We can see examples in human disease brain cells, in knock-in models, and in PC12 neuronal models. Now, this aggregation is important, but I think right now we need to think of it in the context of the no longer emerging view of many proteins engaging in liquid-liquid phase transitions. Numerous proteins form macromolecular assemblies as a part of their normal process. They often have intrinsically disordered domains within them, and they can form dynamic structures that can be liquid-like, and in the setting of oxidative stress or some other stressor or a mutation or a repeat expansion, the expansion of polyglutamine, for example, this more liquid-like dynamic behavior of the proteins may lead to a more solid-like state that can even be fibrillar and aggregation-like. And so I'm just highlighting a nice article, 2 years old now, from Pesquet et al., showing that in Huntington's disease we can see this. There is not a great deal of work that has shown that the various disease proteins engage in liquid-like behavior, but I can tell you our own studies back in early 2000s published in PNAS showed that ataxin 1 and ataxin 3 as full-length mutant proteins formed highly dynamic nuclear structures. We called them inclusions at the time, but now I would say they clearly represent membraneless organelles or membraneless structures that were dynamic in behavior. And when you think about this kind of behavior of proteins, we need to put it in the context of the polyglutamine disease proteins shown here from a very old figure, review figure, that the various disease proteins in the SCAs as well as Huntington's disease, DRPLA, and SBMA are very different proteins. They don't look the same. They don't have the same functions. They're different sizes. The polyglutamine domain, shown here as a yellow band, is in different parts of the protein. But all of these proteins, I will submit to you, are proteins that can engage in transcription products. It's a C-terminal is part of this protein that includes the glutamine domain that is probably intrinsically disordered and goes to the nucleus. I see my internet connection is unstable. I hope you're still hearing me. Are you hearing me?
Yeah, but sometimes you're frozen, so I don't know where you were standing. Okay, go ahead.
Can you hear me now? Can you see me? I've moved the slide.
Fine, yeah, fine.
Okay, so the various disease proteins have different functions and they do different things. Again, we talked about the calcium channel here in SCA6, but in the case of SCA2, ataxin 2 is a protein that is intimately connected to basically translational control in stress granules. And this is a classic case now where I think a modest expansion or modest change in the size of the glutamine domain in the protein is affecting its function in a way that leads to a risk factor for ALS. Can you not hear me? Mathis, you talking to me?
No,
Hello?
please go ahead. Please go ahead.
Okay. Ataxin 1, ataxin 7 are in the nucleus, playing a role in transcriptional regulation. And over here on the right, ataxin 3, my favorite protein, is a protein that's a deubiquitinase, and is shown here very simply doing that function in the cytoplasm and in the nucleus. And the reality is this protein, when there's an expansion, goes to the nucleus and probably is affecting its many different functions in ubiquitin-dependent, DUB-dependent quality control. So this is a review figure from a paper with Drs. Mariotti and Klockeder that shows some of the various pathogenic mechanisms. In the case of the polyglutamine domains, we've already talked about these various interactions that can affect things. And so I won't spend more time on that. I will instead talk about the fact that this is showing in neurons, but we now recognize that these kind of phenomena are not localized simply to neurons. For example, years ago, it was shown by Alla Spada and team that the Bergman glia in the cerebellum are important in SCA7, and my own group, now led by Haley McLaughlin, has been looking at oligodendrocyte role in SCA3. Clearly, we need to know much more about how other cell types are being involved here. And another important point here is I've focused on the protein and how polyglutamine is involved here. It's clear that in some repeat expansion diseases, RAN translation has occurred. Again, in the case of the polyglutamine domain proteins, this would normally encode a glutamine stretch shown as the Q here. But if there is RAN translation occurring here, there can be different frames generating polyserine or polyalanine in the sense strand. These are RAN translation products in the sense strand shown here with the arrow to the right, but also in the antisense direction, you might generate some antisense products. It's quite clear there's good data that in Huntington's disease, there is evidence for RAN translation, and clearly in some other repeat expansion SCAs, less clear so far for the polyglutamine SCAs. So when we speak about RAN translation and its role in disease, I think it's clear that SCA 8, 10, 31, and 36, and probably others, are affected this way. And notice that I've put the SCAs here between RAN translation and RNA toxicity, because I don't think you can separate this out. These larger repeats that in the transcript are probably affecting RNA binding proteins, affecting RNA homeostasis, also can cause RAN translation. Again, principle of parsimony is no longer applicable in the case of these repeat expansion diseases. Now, channelopathies. You heard this morning from Dr. Kenny, and you heard from Dr. Shakate yesterday about the importance of channels in disease. In the case of channelopathies or the involvement of channel dysfunction, directly SCA 6, 13, 15, 16, 19, 22, 41, 42, and 44. But I think the reality is many different SCAs, whether directly involving a channelopathy or involving dysfunction of channels due to aberrant behavior of voltage activity in membrane potentials in neurons, are affected. And I can't go through this in great detail, but I believe this is an area where there needs to be more work to identify these druggable targets. These are very druggable targets that are relevant to numerous ataxias and may be useful symptomatically as well as from a disease modification standpoint. No time really to talk about bioenergenetics, but I think DNA repair is critically important. You heard this morning a beautiful talk showing the importance of DNA repair enzymes in Huntington's disease, and I think the reality is we all recognize that in numerous repeat expansion diseases, there are problems in the nucleus. We know that there are changes in nucleocytoplasmic transport that affect various things. I already mentioned to the ataxin 3 protein tends to go to the nucleus. We know that's true for many, many different repeat expansion SCAs. And I think the evidence for DNA damage and for involvement of DNA damage repair proteins is increasingly clear. I believe that Celeste Suart spoke about this with respect to SCA1 and ATM kinase. And in our own group, Haley McLaughlin is looking at ATM kinase involvement in SCA3, and Nico Dantuma and Partha Sarkar and others have shown that clearly DNA damage repair is affected in SCA3. So I think a lot more needs to be learned about this. And I'm highlighting here the nucleus because we have to go away from thinking about the nucleus as a big soupy structure. It is highly ordered. There are numerous molecular condensates that exist there. Euchromatin and heterochromatin are occupying space in a highly regulated way with various protein assemblies. And when you have a protein that aggregates in the nucleus or forms internuclear inclusions, whether they're aggregates or dynamic assemblies, and when you have changes in DNA integrity, this is going to be affecting the way neurons and other cells are affected in the brain. And let me finish here by saying that we know not nearly enough about the numerous SCAs that are due to more traditional missense mutations, in which in some cases are affecting the behavior of the proteins in toxic ways. Now, my talk is to talk about models as well. This is my single model slide. I just wanted to mention in the case of SCA3, we've been very fortunate that many talented scientists have generated numerous models over the years. We have used transfected cells, mouse embryonic fibroblasts, iPSCs, human embryonic stem cells to look at mechanisms and to test therapies. We've used Drosophila and C. elegans models to really look at genetic pathways in a very rapid way. And numerous mouse models exist, and even beyond that now to other models, whether it's knock-in, knockout, viral delivery, transgenics that can be used. Of course, all of this requires looking at the human disease brain gold standard. And of course, in vitro studies are a critical part of this as well. But despite all of this tremendous overlapping model systems that have been used to look at therapies, we don't have a therapy yet for SCA3. And when you look at the other SCAs, we have relatively few models for many of them. And that's a problem. We need to overcome that problem. I personally think as a field, we need to spend a lot more time right now in an organized way to degenerate through genetic engineering, iPSC lines, or even perhaps human embryonic stem cell lines, because we think there are differences between those two that actually harbor the various mutations so that we can begin to do screening for compounds that might make a difference and explore mechanisms in cell-based models and even human organoids as well. Making mouse models for all of these is going to be something that's going to be a pie in the sky. We can't go there. Yes, we can use Drosophila and nematode to look at these mechanisms, but ultimately we need to look at human cell models as well. So with that background, we now recognize clearly that in the SCAs there's a dominant toxic action in most cases. This means there are many different ways one could actually target the gene products, and I don't have time to go through this in any detail. I will, however, focus on Carmel Costo from our group, very talented assistant research professor, who has done a lot of work on compound screening that has been useful in SCA3 and has been used in other systems as well. In the case of SCA3, and this is true for any of the SCAs, if you talk about the gene product here, ataxin-3, there are many different cellular pathways that might be targetable to reduce levels of the toxic agent, if you will, whether it's a transcript or whether it's protein. And the best way to get at this is not to do a candidate approach, but actually do some kind of a genetic and or compound screen to find potential targets and then move forward for therapies. And so Carmo has done a beautiful job of assembling the various models that we have to actually screen compounds. And she's used in her case, mostly a cell line, a stable cell line that has ataxin-3 with a luciferase fusion so that she can actually screen rapidly for any compound or any gene that actually reduces levels of the disease protein. And of course, to be successful at this, you have to have an iterative screening system that goes from cells to slices from brain to human neurons into model systems, ultimately into mice, to try to find really valid targets that are potentially useful for humans. And in Carmo's case, she's done a great job of screening FDA-approved and other compounds to find that aripiprazole, an antipsychotic, actually reduces levels of disease proteins in mouse models. We have not brought that into therapy, and I think that's a point for discussion. It's not an easy drug to use in patients who have movement disorders, and so that needs to be taken as a caveat. But doing various compound screens, including genetic screens, Carmo has identified other potential pathways. And so, for example, in a druggable genome screen that she recently published in Neurobiology of Disease, she identified, among other things, numerous genes that are tied to TNF and NF-kappa B ERK kinase pathways. These are highly druggable targets, and she's exploring that as we go forward. Now, we don't have a therapy yet, but I think that kind of compound screening is critically important. So in summary, and this has been a whirlwind, and I apologize for that, the SCAs mostly act in a dominant toxic manner via several mechanisms, and they are not mutually exclusive. I'm sure that more than one mechanism is occurring in many of these, particularly the repeat expansions. The models have led us toward disease understanding and have revealed and confirmed in some cases therapeutic targets. While nucleotide-based gene silencing, which is appropriately the fashion right now and holds great promise for numerous SCAs, the search for potential disease-modifying and symptomatic compounds should remain broad, and we need more screens, both genetic and compound. And I think finally, and this is the most important point I'll say, is more work is needed to generate, simple human cell-based models for the full range of SCAs so we can expand this beyond where we are currently. I will stop here. I have a lot of people that I have the pleasure and the opportunity to work with, but I really wanted to highlight particularly two assistant professors, Drs. Costa and McLaughlin, and you've seen Dr. McLaughlin's name earlier today, and I'll stop on that note.
Thank you very much for this brilliant talk like usual. So are there questions? I was puzzled that you didn't say anything about the loss of the normal function of the proteins, which could be very important in the phenotype. And
yeah, I mean, it's partly given the time, the limit, and I realized that I just could only highlight big points. And I think by saying that multiple mechanisms are at play, I should have mentioned that the loss of normal function, it can be a big part of this. In the case of ataxin 3, we know that when you take away ataxin 3, you can see changes in gene expression. So clearly it does play a role there. You see changes in polyubiquitin accumulation in cells, but we don't know that that is really relevant to the disease mechanism. I think more needs to be done there. We don't yet know the answer in the case of SCA3 that a full loss of function would be okay in humans. I personally think that it's a bit player in the case of SCA3 as a contributor to the mechanism of disease. I don't think that's true for other SCAs.
Okay. So I have a look on the Q&A. You said that you have to leave.
Well, I have to see if the residents have patience for me to see, but if they don't, I will come back and I'll hear Dr. Puccio's talk.
Okay, okay. Are they I have a question from Sue. She asked, will you need patients, advocacy groups help in getting the human cell models?
I think it would be
ready to do things.
So it is great to generate cell lines directly from individuals who harbor mutations, and that has been critically important. And thank you to all the patients who have done that over the years, and I have benefited personally from that. But in the era of genetic engineering, we have the capacity to now put mutations directly into existing lines. So I think it's a combination of both. So absolutely, patients are vitally important in this regard, and they also can contribute to the research. But at the same time, we need to engage laboratories that are good at using CRISPR-based approaches to directly engineer the mutations for the various SCAs.